Assalamu alaikum and welcome to our channel. I'm Dr. Hibsa. Today we will talk about the protein docking, in which we are going to cover the topic of protein docking with DNA, protein docking with RNA and protein docking with metals. Or either you can also perform protein docking with nanoparticles. The RNA and DNA protein docking and also the ligand protein docking is very simple, but when any metal is involved in any ligand or protein, then the docking becomes tricky. Same is the case with the nanoparticles, as the nanoparticles contain, most of the time it contains metal atom, so their docking is pretty tricky. So in this tutorial today we will talk about that how you can dock any DNA, RNA, ligand, metal, nanoparticle with the protein. For this we will use the Hdock server, which is a hybrid docking algorithm which perform protein docking, protein DNA docking, protein RNA docking, protein nanoparticle docking, and many more. So let's start. Today we will take the example of 1F7Y protein, which is a protein bounded with RNA molecule. We are going to download it from the protein database server. Just download the legacy PDB format. We have downloaded this molecule, and I'm going to copy this protein plus RNA molecule, and I have made a folder Hdock on my folder, and I placed this protein in this. Okay, this is a protein which contain RNA as well as protein. To visualize it, we are going to use the Discovry Studio. Discovry Studio is an open source software. You can get the academic license free of cost. If you are interested in Discovry Studio, we will paste the link of Discovry Studio installation and use it in the description section. You can get our video from here. Simply go to the file, open, and from the desktop we have a folder 1F7Y, and then open it. You can see this is the protein plus RNA molecule. You can view it like this is the, you can rotate it and you can see this is the RNA and the protein. The white chain contains the RNA molecule and the red chain contains the protein. You can change the display from here. Simply you can switch to the display to different mode, like these are the different protein modes. You can view your molecule according to your requirement. Okay, so you can also modify the view according to your need. Here what we are interested in to separate the protein and the RNA molecule. First of all, go to the edit, not edit, but go to the script and go to the selection and select water molecule. If there is any water molecule, it will select all the water molecule. You can see some dots have been selected and they are become the yellow, and then press the delete button over here and it will delete all the water molecule. We want to be clean our RNA or protein water free. Next you have to go to the again go to the selection, or you can see here, you can see some dots. These are the dots. These are some other heteroatoms you also need to remove. You can simply select them one by one and you can remove them. After deleting these other molecules, you need to go to the script again, then select the ligand molecule. As it not contain the ligand molecule, it contain protein and ligand. Simply go to the protein chains and select the protein chains, and then again press the delete button. When you press the delete button, you will only left with the RNA molecule. Then go to the file and click on the save as and save it as a protein database and name it as RNA. Now we have separated our RNA molecule. Then press Ctrl Z button, the protein will be appeared again. Now we have separated the RNA molecule and saved it separately. Now we need to separate the protein and separate it. For this you can go to this small icon over here, click on it. You will see the different molecules. These are some heteromolecules. These are the protein chain B. We can disable the protein chain B. Protein chain B is your RNA, and then we have the protein chain A. Protein chain A is your protein molecule. So from here, select this B chain, which is, when you select it, it will become yellow. Then again press on the delete button. You will get the protein separately. Now you need to save the protein, save it as, and then go to the PDB format and save it as a protein, as a protein molecule. So up till now we have downloaded the RNA and protein structure, then we clean the protein structure, and then we save the protein separately and RNA separately. Let me show you. If I close this one in the Discovery Studio, open the molecule, and here this is our RNA, this is the separate RNA molecule, and then open again, and then open your protein molecule, protein.pdb. This is our separate protein PDB. So we have cleaned the protein and RNA, and we have saved them separately. This step is very crucial because if you do not clean the protein or RNA molecule, it can cause interference during the docking process. For today demo, we also have another molecule which is a gallic acid, which is attached with a zinc molecule. We have prepared this a metal, like this is a gallic acid. Here in this position we have attached a zinc molecule. This is the basic small example for the nanoparticles. So we will demonstrate that how you can, in one example we will demonstrate that how you can dock a protein with RNA or DNA, and in second example we will explain that how you can dock protein with our nanoparticles. Now in our working folder we have a protein separately, clean protein, clean RNA, and Ga_zinc, which is a gallic acid linked with the zinc. Now we have to perform the docking. You have to go to the Google and type Hdock server. When you type the Hdock server, this is the main server, which is hdock physics phys.husk.education.cn. So this is basically the structure of basically the web interface of the Hdock server. Hdock server is basically a hybrid algorithm which perform protein DNA, protein RNA, metal docking, and many more, template based docking, etc. But the important thing is that first you need to clean your ligand and your protein. Here the first part is you want to upload your protein. This is your receptor which you want to make the receptor. Either you want to make a protein a receptor, or either you want to make DNA or RNA your receptor. In our case, we are going to making a protein our receptor. Go to click on the browse button, and this is our protein uploaded. You can also try some examples, or you can also paste the sequence. If you have a faster sequence of protein, you can paste it. The second part is your input ligand. In the ligand you have to upload either your nanoparticle, either your metal bond ligand, either your DNA, either your RNA. Right now we are going to upload the RNA molecule. Please note that this server only upload the PDB format. Either in both cases, either it is a protein, either it is a DNA, either it is RNA, or either it is a nanoparticle structure. First you need to convert into the PDB format. First you have uploaded these information. You can these are some advanced option if you want template free docking only, or you want to see the multiple docking, like you want a cyclic DNA structure, like which means in which you are specific position C1, C2, C3, or many more, or you can specify a residue like on which kind binding site. If you have a specific residue, you want to check either this residue bind or not, you can bind over here. These are the some optional information. You can put your email ID and your job name, and simply, for example, I'm going to print it as protein RNA job. Okay, and if you like, you can send your email, and then you need to submit. When you submit, it will promote it to the next page, like your job is submitted. It is saying that your job is in queue. In queue means the server is very busy, and when your let's suppose right now more than 100 jobs have been submitted across the world, and when your job has been queued in a row, when your turn will come, it will generate the result. What you can do now, you can simply bookmark this page, and after bookmarking this page, you can save it and then come back later and check either your job or your result has been generated or not. And secondly, when your job is completed, if you have provided the email ID, they will send you the message that your job has been completed. The Hdock server is very simple and quick and easy to use, but there is only problem that they have lot of jobs to process. Sometimes it take 1 day, 2 days, or even 3 days. And the other thing that the Hdock server saves your result up to 2 weeks. If you don't see or download your result up to 2 weeks, your results and job will be deleted automatically from the server. We have stored the job earlier, we run the job earlier, and these are the result. When you refresh your page, and you will get the result something like that. In this way you will get the result. Over here you can see the docking score and binding site. Let me show you that these are some example results from the login. These are our stored job results which we performed earlier. You can see these are the results from our RNA and protein docking. You can see these are the different models: model number 1, model number 3, model number 4. You can see that you can see different logging position: model number 5, model number 1. You can see the result in different kind of rainbow or pure. In rainbow the protein is saved, and in yellow this is your RNA molecule in different format. You can choose the different style: cartoon, surface, or anything else. So these are the position, and these are the top 10 prediction. If you click on these top 10 prediction, it will generate the result, and it will give you the result in the downloaded form. If you go to the folder, you can download the folder, and here you can see the top 10 model of your research. Let me show you the example. Simply I copy this one, the number 1 model, and placed it on my desktop. I go to the Discovery Studio, File, Open, Desktop, and model number 1 PDB. So this is the model number 1 which we have docked so far. And you can see the interaction over here. You can change your interaction from this to this, or from this to this. So you can see your interaction, and you can further see the interactions. You can see the aromatic surface area. You can see the hydrogen bonds. You can see the immobilization surfaces, and many more. These are all options are available in our Discovery Studio tutorial. You can visualize later. So what we did, we have downloaded the top 10 prediction. You can also download the top 100 prediction, or you can download all the predictions this server has performed. In the prediction from the down table, you can see the docking score. These are the docking score. The -253 is better than -243. So if the score is decreasing, it means the prediction is this. This is the confidence score, then this is RMSD score, and these are basically the model prediction. If you click on this model, you will get a table like these are the model table. This is giving you the information that lysine is interacting with 379A of the ligand with a distance 2.043. So you will get all the molecule ligand interface residue and their ligand interaction, then receptor ligand interface residue pair, that how they have paired like 367 and 19B, and so on. You will get the information from here. So this is the Hdock server which provide the results. Similarly, you can use this Hdock server for the metal based ligand. You can also use the Hdock server for the nanoparticle docking, but the only problem is the time. This one, that their server is always busy. You can get your result maybe in 2 days or 3 days, but there is a solution. If you want to perform your docking on your laptop, because this is using the Hdock server for the docking purpose. If you want to, if you don't have a time and you want to perform the docking on your own laptop, what you can do. So let's move on this step. So first of all, we have to go to the main server. When you open this from the Google Hdock server, before going to that, you can use the different kind of FASTA format. For example, this is the example. This is, if you go to NCBI and you can see the protein sequence, you can copy this sequence and paste it. So you don't need to paste anything else, you just need to paste the FASTA sequence. Similarly, if you want to paste any other protein, any other DNA sequences, like this one is, for example, this is your ligand sequence, thus just copy the sequence of DNA, RNA, or even you can type ATT, GC, TT, T, and so on. What is your sequence? You can paste the sequence and then you can submit your job. So this is another way to perform the docking purpose. Now if you want to perform the Hdock server on your own laptop, on this main page you can see this is the download Hdock Lite v1.1 standalone package. You need to click on this. When you click on this, you will be promoted on the next page. You just need to type your information, like here my name is Hamad Ismail, and you need to type your email ID and then type your university name. Then click on the I agree, and then you need to click on the submit button. Then when you will submit the button, you will get an email like this one, and you will be promoted to a download link. This download link will be work for you, and you will get the download software, and you can simply download this software. You can see I have downloaded this software. This is the software basically I have downloaded, .tar.gz file. This is my software. We have downloaded it from this server. The Hdock, basically this is the Huang laboratory, which is basically the University of Huazhang University of Science and Technology, and it is academically free, which means you can use it for the non-commercial purposes, like for academic purposes, for research, for publication, and so on. Now we are going to demonstrate that how you can use this Hdock server on your laptop. But to run this software on your system, you need to make two things. First of all, go to the start and type Windows features, turn Windows features on and off. Click on this icon and then you will pop up to a new window. In this window, you need to click on the Windows subset for Linux and virtual machine platform on. You need to check on these two icons, click on the OK, and then restart your laptop. After the restart, go to the Microsoft Store and search for LTS. On this LTS you will find the Ubuntu LTS 24 or 22. Get any of these app. Either you can get the Ubuntu 24 or you can get the Ubuntu 22. This app can be downloaded and installed in your laptop. Why we need these two things? Because the Hdock.out service is basically the Linux base. It is not the Windows base. So to run it on the Windows, we need to make these two changes. First, turn Windows features on and off, and second, install Ubuntu. I have installed the Ubuntu. This is my Ubuntu 24. I have installed it. When you open it, you will get this black window. This is basically the Linux based. It is not so much difficult. It is very super easy to use. We have prepared command for you and we will guide you step by step that how you can use it. If you are interested in more, we will provide the complete installation link in the video description section, where you can simply install Ubuntu successfully on your laptop. So this was our folder. First of all, open it and you will get the Hdock.out. In Hdock.out there are two files you are interested: createpl and Hdock. Copy these two files, copy these two files or extract these two files into your working folder. So these two files are basically the docking file on which we are going to work. Now in this folder we have protein, we have RNA, we have Hdock, and we have createpl. And there is a one file which we have created. These are basically the command file. We will also paste these commands in the video description section so that you can get it easily. These are basically 4 commands you need to run to complete the docking. The first command is way to access onto your folder. This is your folder, let's suppose on the desktop. First you just need to click on this address bar and copy this command. After copying this command, paste into a file like this one, and we need to change this command according to this one. What you will need to do, first of all, you need to add cd space. After cd space /, in the / you need to type mnt, then again /. From the /, this is the basic infrastructure. After typing this, here is the C. You need to type the C over here, but small c. After typing small c, type again the /. After /, you need to copy and paste complete of this one, but the backslashes are changed with the /. Let me do that. Like, I need to remove this one and add /, backslash removed, /, then backslash removed, /, then backslash removed, /. And we need to create it like this one. So your command is completed. Now this you need to copy this command and paste it in the Ubuntu. When you will place in the Ubuntu, you will see this blue line appear. It means you are in the folder. Now type ls. ls means list. When you type ls, you will see you have command.txt, ga.zb, RNA, createpl, Hdock. These are all the content of this folder you can see, which means you have accessed the folder in the right way. After this, we need to run this command. This command is chmod +x hdock createpl. This command is basically used for these folders to executable, which means they can run in the Linux. Simply you need to copy this command and paste in your Ubuntu terminal and press enter, and it's done. Now the third command is your docking command. You don't need to do this, you just need this part. What this part say? It part say use the hdock function, hdock file, and use protein.pdb and use rna.pdb and produce the output file and the name of the output file is Hdock. Okay, we just need to run this command. Let's say this is our nanoparticle. I change the name to this as a ligand.pdb, and this is protein.pdb. What I need to do, I just need to do change the name. It's ligand.pdb and protein.pdb and output, which means I'm going to dock protein with the nanoparticles, and the name are protein.pdb and ligand.pdb. You need to make it sure that the names are exactly the same with the files present in your folder. So I have changed the name as ligand and protein. Now I'm going to copy this command and paste in the Ubuntu terminal and press enter. Now the process has been started. It said it's calculating the grid for this, and it's saying that it will take about 36 seconds to complete, and the job is running, which means it is running the job and producing the docking results. Now you can see that the actual time consumed is 31 seconds. It has produced the docking results. Now we can go to the folder and we can see that the Hdock.output has been generated in our system. I am using the Notepad++. Let me show you what it is. Notepad++ is a small software which is Notepad++. If you install it and download and simply install it, your file will also look like that. You can open it with the simple Notepad or with the Notepad++. I personally like to use the Notepad++ because it is more handy to use. So these are our results. These are basically the result of these are the protein and these are the ligand. This is the grid spacing and angle step, and initial rotation was 0, and these are basically the axis, and these are axis corrections, and then this row, this one -1, these are the basically your docking score, and these are basically your distance, and these are basically the quality check. It should be the 1. If it is 1, it means your docking score is pretty good. So let me explain this one. The first 3 are translational angle, x, y and z, and the next 3 are rotational component, x, y and z, and then it is a docking score, it is a shape complementary score, and the last one is the contact probability, which is approximately 1. The 1 means good. The docking score is the main key player. The negative value means it has strong bonding. The more the negative, the better it is, which means -103 is better than -102. So it has arranged the result according to the top poses. So we have generated the docking score. Now we are moved toward the poses. So in our command list, the last command is to generate the docking score, models, in which I have provided the link. It is going to use the command createpl, and it is going to use the score of Hdock.out, and it will generate top 10 PDB structure and name it as model. So we are going to use this command, copy. Now I close it and open it as a side menu, and then let me shrink it for you. So that you can see the real progress. This is our folder. In this folder I am going to run this command. The command is create top 10 poses with the Hdock. Just look into this folder when I run this command, and now it is running. Now it is now generating different poses. These are the top 10 poses it has been created. To see the poses, you can go to the simply Hdock server and model, open in the Discovery Studio, go to the desktop. In the desktop we have Hdock. In Hdock we have different PDB models. You can open any model in it. Okay, it's a header record. There is something as it is the Linux based model and we are going to open it in the Windows. It has some problem. For this you need to right click on the model and open it with the notepad. After opening it, it says you just simply, you can say this is the docking score and other information. Just simply delete this part and save this file. After doing this, go to the Discovery Studio, open. Now open model number 1. You can see this is your model. You can see this is your protein, this is your nanoparticle structure, and these are basically their models. You can see their models. Again, you can see their interaction diagrams, aromatic interactions, and some other molecule interaction. So you can visualize these top models using Hdock score. So these are basically the result of Hdockings. In the same way, we have performed the docking with the protein with RNA. Let me show you. These are different model I have generated. First, let me fix it header. Open it with the notepad, and I'm going to remove. You can see these are basically the information, and these are the RMSD value 0.82, and these are basically headers. What I need to do is to remove the header, because this header is the main problem. After removing the header, I am going to open it in Discovery Studio. Hdock model 1, open. So you can see this is the basically protein and DNA molecule which has been interacting with each other. These are the docking poses from the RNA and the protein. In the same way, you can perform this with the DNA and the protein. You can also view the result using the PyMol. What information is needed? As I told you earlier, the information you need is basically the docking score, which is basically the out score, then the RMSD value, like this is the main key information you need. This is the docking score, and this is the your RMSD value. From this score you can pose your structure, and you can find out the binding ability. So the DNA, RNA, metals, nanoparticles, all can be docked easily using the Hdock server. You can perform this on the online web server, but for performing online server you need to wait for 1 or 2 days for the result. But if you want to perform it on your laptop and get the quick result, you can perform in just 1 to 2 minutes. But you need to download the Ubuntu and need to follow all these steps as I have demonstrated in this video. So this is all about today. If you have any queries, you can ask in the comment section. Thank you so much for watching. Please like and subscribe. Allah Hafiz.